Really happy to be here, and this is Junbiao from Tsinghua University. And actually, I'm moving from Tsinghua to the Center of Synthetic Biology Engineering Research at the Shenzhen Institute of Advanced Technology at the Chinese Academy of Science. And today, what I'm going to tell you is one story, because in the past several years, we have been always asked why you need to synthesize a genome, right? As Jeff mentioned also in this morning, why we want to do that. So hopefully today I'll give you a short overview why I want to do that. And the topic of today's talk is unlock the power of SCRaMbLE with rescues. And that is a reporter system we actually designed to use the synthetic yeast and to do some applications. As we all know, the synthetic yeast project is an international collaboration project. And within the project, my group is actually working on one of the chromosomes, the chromosome 12, and we synthesize it and by different strategies, and eventually published this year. So the next question is, okay, now we have that yeast strain with a synthetic chromosome. So what can we do with it? As we all know, there's one important feature in the synthetic yeast project, which is a SCRaMbLE system. In short, SCRaMbLE is just a lot of LOXP sites which was integrated into the synthetic chromosome. The presence of the LOXP sites allows us to really, SCRaMbLE or make a lot of variation of the genomes if we induce the recombinase. That is, from a homogeneous yeast population, if we induce the recombination, and now we will eventually generate a population of cells. For each of them, they will have a different genome. So from that population, we can actually select or identify many strains which we are interested. In order to do this, so this is one illustration that's saying that, if we induce the recombinations, you'll see most of the cells are dying, because you can imagine, because there's so many LOXP sites within the genome, and they will eventually hit the essential genes, allow them will not be able to survive. And also, however, to use the SCRaMbLE system is not so easy, because if we induce the recombination, most of them will be dead. And within the survival cells, some of them are actually not SCRaMbLed. They are still intact cells. So in order to overcome these problems, what we did, another thing is that in order to select those SCRaMbLed cells, what we did previously is trying to look for those small colonies, which is an indicating of the genome gets SCRaMbLed. However, this selection is not very reliable, because we can select a lot of the cells, and most of them cells like just gross phenotype differences. And in addition, what we did in the previously is trying to look for the loss of specific markers, as the one showing here. This is MAT17 markers within the chromosome 12, and we did a SCRaMbLE, and we placed the SCRaMbLed cells on a media, contain selecting media, and looking for the loss of the marker. However, the efficiency to get that gene get lost is very low. So in order to solve this problem and to select or identify a lot of those SCRaMbLed genomes, what we did is we tried to design a rescue system. So if we look at the LOXP sites we used the symmetric LOXP site as shown here. And from this literature, we can see that this LOXP same site is orthogonal to the LOXP site. Therefore, based on this literature, we designed a rescue system, the reporter of the SCRaMbLE system, using the LOXP sites. We insert these LOXP sites, franking the two markers with these LOXP sites, and put that into another chromosome location within the synthetic cells. So if there is a query expressed within the cell, and the recombination happens within the reporter system, it will switch the two autotrophic markers, and therefore you'll get loop plus colonies, and they won't be able to grow in your cell. And since there are many of the log P sites, symmetric log P sites on the synthetic chromosome, therefore, once we have a clone which has a LU expressed, that means the log P, the recombination happened at this loci, you can imagine there might be also same or at least one or multiple recombinations also happened on the synthetic chromosome. Therefore, using this method, we can ensure every clone we picked up on this plate also contains some rearrangement on the synthetic chromosome. That leads us to get a lot of those scrambled cells, and from those cells, we can identify the one we want. So, So we use that as a test, and initially we test if this system is tight enough, and it is actually, if you look at this one, showing here, without induction, you'll see nothing grows, and only you induce this for 24 hours, you'll see those LU colony appears. And this is just, induced different times. You can see more of those LU plus colonies appears. So using this system, we actually scramble the chromosome 12, and then using, pick up those rearranged chromosomes, the cells, and as shown here, we pick up the 5 different strands and do a genome sequencing, and to identify all the arranged regions within the synthetic chromosome, we can see all sorts of the modifications like the deletions, inversions, and duplications. However, compared to the one published paper using the circular chromosome 9 arm, and we found that there are actually, for these linear chromosomes, the number of rearrangement is actually much reduced. And we also found a large, inversions, as shown here. It's about 300 kb long. And then using this system, what we did is we actually do a proof of principle experiment to select clones which are able to grow in on the media contain 8% of the ethanols. And we identify clones, and we test its growth in the ethanol-containing media, and they are growing better than the wild type or than the original strains, as shown here. And also there's another clone which are more tolerant to higher content of ethanols. And those clones are actually, produce more ethanols. And then, so this allows us to identify those clones. So the next step is, can we, using the system, to identify why those clones are able to resistant to ethanol? So we are using one method, as we also reported recently, the POPM method, and we cross the ethanol-tolerant strain with a wild-type strain, and then do a sporulation and identify the spores which are resistant to ethanol and which are not resistant to ethanol. And then using a PCRtag analysis, we are able to identify which region of the synthetic DNA are actually correlate with this ethanol resistance. That allows us to identify the region exactly where it is. And of course, we also did a whole genome sequencing, which allows us to firmly confirm that in all of the ethanol-resistant clones, they all contain a rearrangement at this region. Okay, this is involved in one of the LOX-P mediated rearrangement at one gene close to the ACE2 genes. And obviously, then we tested it because it's possible it's defect due to the ACE2 or the RT2. And then we look at the transcription in these strands and looking for if the ACE2 will be affected or not. And it tells us, yes, the ACE2 transcription is affected. Once you have the arrangement happens, it will remove its 3' UTR, which allows to the transcription goes down really dramatically. And we tested by Western blot to check if the protein level is also affected, and it is. So therefore, that leads us to see, okay, this ACE2 are probably the reason for the ethanol tolerance. And then we also tested, if we completely remove the ACE2 genes, and they will confer the ethanol resistance as what the string we identified, and it is. And then that result shows us, okay, we can using the SCRaMbLE to identify the ethanol resistant strains. And we can also, using the method we developed, trying to map where are the resistance they come from. So which is the mechanism of this resistance. And we also probe and trying to understand or using the synthetic chromosomes to see, is that possible to improve the chassis with the SCRaMbLE system? So we actually integrate the beta-carotene pathways at another chromosome location and to induce the SCRaMbLE systems and then select the strains and trying to get some of those strains which are producing more of the beta-carotene, as shown here. And of course, we screened and we get quite a few different strains which are producing more of these beta-carotenes. And we are in the process of to identify why and what's the reason causes those clones producing more of those beta-carotenes. And so this is one example. And we can also, using the SCRaMbLE system in combination of the rescue selection system, to looking for some of the populations which are able to evolve more faster than the original strains. So this is just one example. We were putting the rescue system into a strain, and in that strain, there will be leaky expression of this cree. And then we were putting a selection on that population. And once you have that leaky transcription, you'll see the cells population we got, they can actually grow better under these stress conditions. And now we are also in the process in trying to identify why this population comfort these salt resistances. So I'll stop here and thank the person who did the work. And there's one graduate student, Jiuqing Luo and Li Hui, they did most of the work. And Shen Yue and Yun Wang from the BGI, they did the sequencing analysis and also the funding sources. I'll stop here and take some questions. Good. Oh, no.
Well, it's not exactly a question, but more like a comment.
Mm-hmm.
I think it can be really interesting to take some aspects of the previous talk where you have the idea of relocating like thematic genes. Let's say in that case they were tRNA genes, but you can imagine biosynthetic genes that are involved in a metabolic process or cancer genes or whatever, and relocate them to neochromosomes and then have the SCRaMbLE acting on the neochromosome alone, and then putting that through a series of phenotypic evaluations to, let's say, saturate that particular process and understand it more deeply. With the cancer topic, it could be really interesting. Well, in this case, in yeast would be like cell cycle genes, but it could be really interesting because you would have like the classical amplifications or deletions, inversions, but you would have many combinations of those kinds of events, and maybe we can learn something new.
Yeah, that's brilliant idea. I think, we actually doing something similar, but not in the metabolic sites. We are doing some essential genes using the similar things. Yeah.
Okay.
But yeah, that's a very nice idea. Okay. So, thank you.